We are now being recorded. So welcome to video teleconference number 9 for BIOL 105 YC. This is our last video teleconference before the semi-final exams. So if we look at the schedule, um, we are actually on the semi-finals week already. No? So if you have a subject that's quarterly, then this is your midterms week also. However, for our subject, I will be opening your exam on Wednesday next week. That's March 31. Okay. And what will be the coverage? Chapter 7 as shown here. So we have chapter 7, we have chapter 8, and chapter 9. We won't be including this genetic information in cells portion. In fact, this is actually relegated to genetics. So our next lesson will not be about genes again. Instead, we will be talking about the nucleus. So that's going to be next week. So next week will be for semifinals, no, on Wednesday. Let me admit someone first. All right. So that's for the calendar, no? So March 31 for the exam. This morning, we're going to be reviewing the last topic, no, that's included in the semifinal exam, and that's chapter 9, the cytoskeleton and the cell motility. Okay, so let's start. Okay, so um, the cell, just like vertebrates and even invertebrates, now have a system of um, skeleton on the inside that we call the cytoskeleton. So as shown in the illustration here, we have three examples of cells showing their cytoskeleton. We have an epithelial cell here, we have a neuron here, and then we have a dividing cell here. So anyone from the group, choose a cell and then just highlight, no, summarize the function of the cytoskeleton in that particular cell.
In the epithelial cells, sir. So they the cytoskeleton they provide its structure and support as seen in the actin filaments, which it allows it to be an upright position, and also the microtubules na makakuan siya into upright position as well, and the intermediate filaments which connects the cell on each other, sir. Especially the desmosomes.
Thank you. Okay, so you're correct now. This portion here, the one that I'm pointing is a desmosome. Also, this one here on the left is a desmosome. And then the actin filament, if you're not familiar now, this is actually a cell from the small intestine. So these finger-like projections are microvilli. So they are held in an upright position, as you mentioned, because of the actin filament support. And also the overall columnar shape of the cell is maintained thanks to the microtubule, okay, that we see here. Okay, so thank you for highlighting those. Okay, another cell, please, from anyone.
For the neurons, or it has these actin filaments, which um, enables for the formation of branches in the neurons. There's also the microtubules and the intermediate filaments for structure and support, and it also has motor proteins which engage in intracellular transport inside the neurons, sir.
All right, so those are correct, no? So actin filaments, as you first mentioned, allows for the growth of projections, no, from the neuron. So it's very important for the neuron nga magsanga-sanga, di ba? So the actin filaments make those formations possible. No? So anything that you see, the extensions in a cell, so for example, an amoeba producing the false feet, no, the pseudopodia, pag may extension, those are usually begun by the formation of actin filaments. And then, of course, the intermediate filaments and microtubules here, this is actually the axon of the neuron, serve as the main skeletal structure of the axon for support. And most importantly, as you mentioned, the microtubules here serve as a pathway for the motor proteins carrying vesicles. No? The vesicles here might contain neurotransmitters that needs to be brought to the synapse, for example. That's also true for the epithelial tissue that was mentioned a while ago. So you can see here also motor proteins using the microtubules as pathways. All right. So we have this last cell. Anyone? Actually, two cells. Two cells here at the bottom.
This one, sir, for a dividing cell, it's more on ano here, for the intracellular transport. I think these ano, sir, mga microtubules, they are attaching to the chromosomes, sir, and then they try to somehow pull away from each other. So, they're or na-transport nila kahappen sa microtubules as they grow. And then the formation also of cleavage furrow sa mga ano, caused by the as the actin filaments here, sir, are contracting. Somehow they act as ano, rubber bands to like pinch the cells off from each other as they divide.
Right. So those are correct. So again, no, there's another name for these special type of function for the um, microtubules. These are actually known also as the spindle fibers, no? So you know them as spindle fibers. So spindle fibers are microtubules that help the sister chromatids separate from each other, no, during mitosis or meiosis 2, or they separate homologous pairs, no, during meiosis 1. Um, we also have here in animal cells the formation of the cleavage furrow, as you mentioned, is um, facilitated or helped by actin filaments. And then very important also, we see in this particular illustration, not these organizing centers of the microtubules, two of them. Also seen here in our epithelial cell, no, on the left. There's also an organizing center. We'll talk about that in the next slides. All right. So the cytoskeleton is composed of three main components. No, there are three main components. So listed here, all of their characteristics. So anyone from the group wants to highlight a similarity or differences between these components?
For differences, sir, I can say that microtubules and actin filaments, uh, titawagan ni, their preferential site of incorporation or nucleation is on the positive end for both, and for intermediate filaments, they are more internal and like more random, sir, in nucleation, sir.
All right, so that is correct. So you're about to mention, that there's polarity also, in microtubules and actin filaments, and the nucleation is on the plus end. While in the intermediate filaments there is no polarity, and the addition or removal of components would be random, no, or even internal. So that's one difference. Okay. Anyone else? Any other differences or similarities between the three?
Their structure, sir, as seen from the cotton illustration a while ago, ang microtubule is stiff, hollow, inextensible tube because it's where the motor protein um is a pathway, and actin filaments, as seen in the epithelial cell, it is flexible. Then ang intermediate filament is tough, sir, although it is also flexible.
All right, so thank you. I'd like to highlight what you said on motor proteins, no? Okay, so take note that microtubules and actin filaments are polar. They have polarity. There's a plus end, there's a minus end. So this allows them to serve as guiding for the directions of the movement of the motor proteins, no? Because you mentioned motor proteins a while ago. Well, notice in the intermediate filament, there is no polarity, and therefore they cannot guide a motor protein, no, in which direction it should move. So there are no motor proteins involved in intermediate filaments, but there are for microtubules and actin filaments. And then you also mentioned structure. Um, Later on, now we'll see more of that. But this table is summarizing naman. So later, we'll see instead of just reading the words about their structure. So I'll talk about that later. So thank you for that. No? So another one, siguro, last observation on similarities or differences between the three.
For the enzymatic activity, sir, both the microtubules and actin filaments have it. It's just that some microtubules is GT-PACE and as actin filaments they use AT-PACE. While on the intermediate filaments, they have no enzymatic activity involved. And then also, sir, in intermediate filaments, it is also found in the nucleus. And the microtubules and actin filaments are like inclusively only in cytoplasm.
Right. So thank you for that. So thank you for pointing out the enzymatic activities. No, very important to remember. What does this mean? Sa mga GT-PACE, AT-PACE. This means that for microtubules, in order for it to elongate, di ba? So later we'll see an illustration of how it needs to use GTP as a source of energy, not to elongate microtubules. For actin filaments, it needs to use ATP, not GTP, in order to elongate. And then for intermediate filaments, they don't even need to use ATP or GTP to add or remove components from themselves. No? And then thank you for pointing out that intermediate filaments are actually found also in the nucleus. They actually line the nucleus. Um, the lining is known as the nuclear lamina, and it's made of intermediate filaments. And I don't know also, was it this group na nag-mention that intermediate filaments are keratins? And I think I sort of mentioned that no, they're not keratins. I was wrong. Okay, intermediate filaments are actually the keratins, no? That help in structural integrity of the cell. So keratins. All right. So thank you for that. No. So what else can we see from here? Their dimensions, no? The thickest will be the microtubules. Intermediate filaments follow, and the thinnest are the actin filaments. That's why they're also known as the microfilaments, ang actin filaments. And then in terms of distribution among living things, intermediate filaments are only found in animals, no, animal cells. So not in plant cells. All right. So um, let's move on to the next slides because we are going to detail there the structure and the other things about these cytoskeletal elements. Starting with this one. Okay. So anyone who wants to explain what we see here on this slide?
It is a microtubule, sir. Then it is composed of 13 13 alpha and beta tubulins. Then it is rod-like in shape and it is hollow, sir.
Right, so thank you for that, no? Just to clarify, it's composed of 13 protofilaments, no? So kaning galing niya nga beta, alpha, beta, alpha. Kaning siya nga one line, one vertical line is a protofilament. So if you're going to count how many there are in one roll of microtubule, there are 13, no? What else? Anyone? Unsa pa ang ma-observe ninyo? Kaning nasa right, what's this?
On the right, sir, it shows um, the actual structure of the alpha and beta tubulin, wherein the alpha tubulin has a GTP subunit and the beta tubulin has a GDP subunit. And also for beta tubulin, it is the positive end of it is on the positive end of the microtubule, while the alpha tubulin is uh, forms the negative end.
Okay, so thank you for that. So this whole thing, kaning beta and alpha tubulin, they always come together, ha? They're always together. You cannot find them separate na, oy, beta tubulin lang or oy, alpha tubulin lang. They're always stuck together. And together, they are the monomers of the microtubule. No? So, ang monomer sa microtubule, always gauban permi ang beta and alpha tubulin. Always. Okay. What's the reason why this is the plus end and this is the minus end? Notice you mentioned no the GTP and GDP present in alpha and beta tubulin respectively, no, shown here. These are not subunits. Okay, ang subunit is a portion of a protein talaga. No, these are sort of accessory portions of these molecules. So, bakit plus end ito? Notice this is GDP. Why is this GDP? And why is this GTP? Okay, that's my question. Anyone knows or has a guess why?
Due to the presence of GTP in the alpha tubulin, sir, it is more negative because it has an extra phosphate group, which makes it more, which makes its polarity more negative, sir.
All right, thank you for that guess. No, you're guessing. This has nothing to do with charges. Itong minus plus, no? These have nothing to do with charges though. But thank you for that. No, there's a logic in that. Any other try? Why is this GDP and why is this GTP or why is this minus and this one's plus? What can Okay, continue.
GDP can be found in the beta tubulin, sir, because it is a GTPase, which means it hydrolyzes GTP.
All right, nakat ka no, pero I got what you said. GTPase, remember in microtubules, is the enzyme that helps polymerize the microtubule, no? And the polymerization can only happen on the plus end. Now, it can never happen on the minus end. Why? Because the GTP in the alpha tubulin portion is sort of hidden, no? Naa siya sa sulod in between the alpha and the beta. While the GTP in the beta tubulin is near the tip, tanawan no? Mas exposed siya. So it's easier for the GTPase to hydrolyze it to become GDP. Mao ning gipakita diri GDP because it has already hydrolyzed. Okay, thank you for that term. And that hydrolysis is coupled with the polymerization of new alpha and beta tubulins no on top of this. So mao nang plus end siya kay dito magdugang, that's where you add new components of the microtubule. All right. So notice also, is that clear guys? No, nganong plus end? because that's where the reaction can happen. Okay, again, nothing to do with charges. Okay, but you can always rationalize on that. Another thing to note here is the asymmetric way of arrangement. What does that mean? If you look at here at the bottom, the ending is alpha. But if you look at here on top, the ending is beta. So it's asymmetric. No? So there is really polarity. Another thing to note is the seam. No? The seam. Okay, I need to admit someone first. Unsa man ang seam nga makita nato dire? Okay. I think the easiest way to visualize this or imagine it is if you're going to fold or roll a cartolina, no, nakatry na mag-roll sa cartolina, then there is going to be that seam where the two edges of the cartolina will meet, di ba, when you roll them. And it's hollow in the middle, just like this microtubule. So imagine a microtubule as a rolled cartolina or a rolled paper. Then we have this illustration. What is shown in this illustration? Anyone?
It shows the motor protein, the two different motor proteins associated with the microtubules, sir. For the one in the bottom, it's the kinesins, which carry vesicles towards the positive end of the microtubule. And the one on the top are dyneins, which carry vesicles to the negative end of the microtubule.
All right. So thanks to the polarity of microtubules, they are able to guide motor proteins. And there are two types associated, no? The dynein and the kinesin. So the kinesin can only move in one direction, no, from negative to the plus end. So if ever a vesicle needs to be moved backwards, then it has to be the dynein that will do that, no, which can move backwards. So in terms of structure, do you still recall the differences in terms of the characteristics of the dynein and the kinesin? Anyone?
In terms of the kinesin, sir, it has four main components. The one that attaches to the microtubule is the head. It also has the neck portion, then the main stalk, and the tail, which attaches to uh, an adaptor protein, which helps it attach to the vesicle, sir. And the tail is made of a there's a lighter chain found in the tail, and the heavy chain is found more on the stalk. Stalk of the kinesin, sir.
Okay, thank you for that detailed description. Okay, so that's correct, no? Another thing to note is that dyneins are a lot bigger, no, than kinesins. Okay, but it doesn't really affect on the strength, no, of these two motor proteins. Dyneins are also much conserved, meaning to say there are very few types of dynein. In fact, there are at least two, or at most two of them. There's the cytoplasmic, and then there is the flagellar or the ciliary dyneins. While for the kinesins, there are many types of kinesins, no, as mentioned in the video that I posted. In terms of energy source, question, what energy source do these motor proteins use? Is it GTP or ATP?
ATP, sir.
All right. So it's always ATP that's used by the motor proteins, no? Don't confuse that for the GTP. The GTP is used for the polymerization of new microtubules in the plus end. Mao na ang GTP function. All right. So, and then here we see several cells and we see the structure, the ones drawn in blue in these cells. What's common between these blue structures?
Their charges, sir.
They are charges. The
No,
charges are,
sa sa nucleus and go siya outward from the cancer, from the nucleus.
mhm. Just to be clear, the structures, their position is not really related to the position of the nucleus, okay? But thanks for trying. Although they look like they're the nuclei here or they're near the nucleus, but they're not, okay? They're not associated with the position of the nucleus. So thank you for trying. What are these?
These are centrosomes, sir, wherein uh, this is where the sir, microtubules form, sir. Form from, sir.
All right, so the ones on top are centrosomes, no? These are centrosomes, and you can see microtubules forming from them, as you mentioned. So the minus end is near the centrosome, and the plus end is away from the centrosome. No? So look at all the cells. Ang minus is really towards the centrosome, while the plus are away from the centrosome. No? So the red arrows are the microtubules. Um, this means that they help in organizing the positions or the orientations of the microtubules. Sir, they are also known as the organizing centers of microtubules, no, or MTOCs, microtubule organizing centers. And we see here in the middle a pair of structures. What are these?
The centrioles, sir.
All right, so these are centrioles. No, centrioles are um, actually only present in animal cells, no? Or maybe I'm mistaken, maybe it's a centrosome. But these are only obvious, ganito na lang, they're only obvious in animal cells, no? And they cannot be observed in plant cells. So that's for the centrosomes. What about this one at the bottom? So this is
That
a
is
sperm.
the basal body found in sperm cells, sir.
What is its function?
It functions in also in the formation of microtubules uh, for the tail of the sperm, sir, so it can be motile, sir.
All right, so the basal body is actually similar in structure to the centrosomes, no, that we see here on top. And therefore, it is also an organizing center. No? This is also a microtubule organizing center. So you can see microtubules growing from it into the flagellum. So the minus end of the microtubule is at the base of the flagellum, while the plus end is at the tip of the flagellum. Okay. And then here we have a detailed look, no, at one of those, the centrosome. Okay. So what do we observe in this illustration? Anyone? What is this blue structure at the bottom? Because this is a centrosome naman. So what is this specific portion, this blue structure?
This is the blue structure at the bottom, sir, is the pericentriolar material, sir, if I'm not mistaken. And
Yes.
on the middle of it is the uh, gamma tubulin uh, ring complex, or gamma Turk. Yeah, gamma Turk. And it functions as a nucleating center for the microtubules, sir, for the alpha and beta tubulins to form on, sir.
All right, thank you. So remember, microtubules are made of alpha and beta tubulins, not together. There's actually another type of tubulin called the gamma tubulin. And the gamma tubulin can only be found embedded in the pericentriolar material of the centrosome. No? So it's here at the bottom, no, kani murag ring that is shaped like the shell of a snail, and it's attached to proteins. No, it's held by proteins. So all in all, this one is collectively known as the gamma tubulin ring complex, and it is where the alpha tubulin, no, in the dimer attaches first. So kani mag-attach, no. So this serves as the attachment for the minus end of the microtubule. And then once that is there, that's present, the one that is exposed will be the plus end, the beta tubulin portion. And so new alpha tubulins can attach, and then on the plus end again will be their beta tubulin subunits. And this can continue on and on, no, until the microtubule is long enough, no, for the purpose of the cell. So as you mentioned a while ago, that process is called nucleation. Nucleation, the formation or the polymerization of microtubules onto this gamma tubulin ring complex. Okay, so this is a detailed look of that. So that's why it's called the microtubule organizing, no, center, because it helps orient the microtubules. And this one, can we see any obvious centrosomes or centrioles in this illustration or in this slide?
Wala siya, sir.
Wala, no? So, but there is still a gamma tubulin complex, but it is along this microtubule. So we have a parent microtubule, now shown in this electron micrograph. So you have a parent microtubule, sort of like a stem of a plant. And then along that parent microtubule, there are gamma tubulin complexes, sort of like the nodes, no? Nodes of a plant stem. And from these gamma tubulin complexes, new microtubules can form. So morning and then magsanga-sanga nga microtubules. And yes, there are no obvious organizing centers here. So this must be in a plant cell. No, sa plant cell, wala sila obvious nga pareha ani nga naay centrosome, then naay pericentriolar material, wala. Sa plant cell, the gamma tubulins are distributed along a parent microtubule. So here at the bottom, we can see a new microtubule growing. No? So here it's elongating. And it's originating from a tubulin complex on a parent microtubule. So that's in plant cells. And then here we have an illustration, okay, regarding microtubules. Can anyone from the group tell us what's happening here?
This is the concert dynamic instability of assembled microtubules. This is where, due to unstable positive ends, or uh, the positive end, of course, will break down eventually, sir.
All right. Sige, so that's correct, no? We see here in this illustration na that kaning pula na mga portions, no, these are alpha beta tubulins. They still have their GTPs, no? But as they are incorporated in the growing tubulin, they close, no, musira. Then ilang color color coded na himon din sila blue. What does this blue color mean? Meaning to say they're already used up their GTP, no, in the positive end, no, in the beta end, and those have become GDPs. So morning na blue na dayon. So kaning red, ang nasa beta is still GTP, but with the help of GTPase, it will be converted into GDP and the microtubule closes here. However, there is this random or spontaneous disassembly from the plus end, no? So I would like to liken this, di ba? A while ago, we said that the microtubule is hollow. It's like a cartolina that you roll. Imagine rolling a cartolina. No? So you roll it, and then you let go. What's gonna happen?
Mabungkag siya, sir.
All
Because there's
right. So
nothing to support it.
yes. So in the same way, a microtubule that is formed will experience catastrophe if there is nothing to support it in the plus end. So sa cartolina, unsa man atong himuon, we put tape, okay, to hold the two edges together. But in the microtubule, what is the equivalent of the tape?
Um, they can there can be the attachment of plus tips, sir.
All right, so this is where the microtubule associated proteins, no, or MAPs come in. And a special type of MAP is the microtubule, is the plus tip protein. The plus tip protein stays here in the positive end and help stabilize our microtubule so that it will not be disassembled like this. Now, so kinahanglan siya og another protein called plus tip, which is a type of microtubule associated proteins. Okay, so ganun lang. So just remember that one. And then here we have the microtubules as seen on how they are arranged in a cilium or a flagellum. So notice here at the bottom, here's the basal body. And then here is the cross section of the basal body. What does this resemble?
Parang siyang centriole, sir.
All right. So remember the centriole kato illustration ganina sa sperm and the other cells. The basal body is basically just the centrosome but in the base of the flagellum or cilium. So they are the same. They are both organizing centers. And we can see in the arrangement here, these are nine, no? Nine spokes. And then here we have three microtubules. No, we have A, B, and C. Question, are all of these microtubules complete or no?
No, sir. Only the A tubules are complete, sir.
Okay, correct. So, kaning A tubule is the kaning pinakasulod, no? You see it's complete circle, but if you go out, this one is not complete circle and the last one is also not a complete circle. So that is also translated here in the cross section of the cilium or the flagellum. How do we call this again? This structure starts with letter A. Kaning
Axoneme, sir.
axonym, no, axonym. So this is the axonym. So you can still see here, no, nine kabuok spokes, and then we can still see there are still microtubules, but instead of triplets, they are now doublets. And we still have the A, the complete microtubule, no, the dark blue, and the B, the light blue colored microtubule here. Okay. And notice from the A microtubule, you can see those small structures that are extending outwards towards the B of the other doublet. Okay. So you look at the A, there's always these small stuff sticking out, now pointing towards the next B microtubule of the doublet. What are those? What are these? These extensions from the A microtubule.
Dynein arm, sir.
Yes, all right. These are dyneins, no? These are going to make contact with the B of the next doublet, and with the help of ATP, it's going to push that doublet in a certain direction so that the cilium or the flagellum will move. No, so um, if we're gonna go here, no, we see the two doublets shown. So we have the B here, and then we have the A. From the A, we have these dyneins, no, and then the dyneins make contact with the B tubule of the next doublet, and then with the help of ATP, it's going to have a power stroke. No, it's going to push this doublet so that they're going to move sideways, and then it's going to detach and get ready for another power stroke, and then the cycle continues. So as that happens, your flagellum or cilium actually moves from side to side, as we see here. So that's how the dynein in the flagellum or the cilium works, with the help of ATP. Okay, and also shown here, in the previous slide, how materials from the cell are carried to the tip of your cilium or flagellum. No? What does that? Is it the dynein or the kinesin from the cell to the tip?
Kinesin, sir.
Kinesins, no? Because that's the tip is the plus end, di ba? So look at this illustration at the top, no? So from negative to plus, that's the kinesin that will carry the material to the tip of the flagellum or cilium. But going back, of course, from plus to minus, that will be the dynein. No? And this one is a special type of dynein. This is cytoplasmic dynein. Kaya iyang role is to carry vesicles man. But a while ago, kanina dynein, this is not cytoplasmic because this is attached to your alpha tubule. No? So this one is the flagellar or ciliary dynein. All right. And then we have this illustration. So this one is for the intermediate filament. Can anyone from the group point out which among these is the basic subunit of our intermediate filament? Is it number 1 here on the left, number 2, number 3, 4, 5, 6? Aha man ang basic subunit sa atong intermediate filament dire.
The third one, sir, the tetramer.
Okay, how do you know or why? Okay, in order to answer that, you go back to the characteristic of the intermediate filament. Di ba? We said that it doesn't have polarity. Wala siya plus end or minus end. So you look at these structures. Which among them is the smallest with no plus or minus end? No, dili man pwede kaning monomer kay naman siya N ug C, no? They are obviously polar. Ang dimer po dili pod pwede kay na may N ug C. So the next, the tetramer, you notice ang ending niya diri sa ubos is C. Ang ending pod niya sa taas is C terminus. So therefore, it's the first or the smallest among these that are nonpolar. So therefore, it's the basic subunit of your intermediate filament. Okay? And notice here on the right side how the polymerization is like in the intermediate filament. There is no plus or minus end. So it can just happen anywhere. You can just add a new subunit here in the middle or remove one also here from the middle, no, for the intermediate filament. And of course, these are keratin, no, made of keratin. So in summary, we see here a cell with the cytoskeletal elements. Okay, can anyone just say a summary of what we see here? Anyone?
For uh, the formation of cancer, microtubules is can be found here in the mTOX, or the microtubule organizing center. So yeah, the microtubules are nucleating from that, and they provide the structure of the cell, and also serve as pathways for cellular transport. And we can also see the ones in pink, which are the intermediate filaments. They are attached also into desmosomes and into hemidesmosomes, to provide structure and support for the cell. And there's also these yellow ones, the actin filaments. And on the edges of the cell, they form sort of little projections on the edges, sir. And also, they are also attached to adherence junctions, sir.
And focal adhesions, no? Katong,
Sir, focal adhesion.
so here, what is this green molecule?
That is integrin, sir.
All right, so these are integrins. So see, this illustration is sort of a includes also our topic on cell to cell adhesion, no? Or cell and surface adhesion. And another thing to point out is we also see some pink here inside the nucleus. That also indicates the presence of intermediate filaments, no, lining the inside of the nucleus, no, the nuclear lamina. Right. So the last component of our cytoskeleton of the cell are the actins. No, that's the one that we see here. And if we describe the structure, it's just really alpha helices ng actin. And then this is one monomer, kaning isa ka blob. That's one monomer of the actin. Then look at the structure. There is a plus and a minus end. So this time, it's the ATP, no, that serves as the energy for the polymerization of our actin filaments. And we see here an electron micrograph, and if we have a clearer view of that, you will see that it actually is shaped like arrowheads, no, attached next to each other. So na ay sharp end, na apoy barbed end. And noted here, the barbed end is the plus end. That's where the growth of the actin filaments is faster. While in the minus end, that's the where the growth is a lot slower. Okay, so what's the difference between the growth in the microtubule and actin filament? No, I just mentioned in the actin filament, it's possible to also grow in the minus end. So look at this. Naagihapoy gamay nga nitubo from the minus end. But in microtubules, it's really impossible to do that. No, some plus end ra gyud. So what the detail of that is this one. No, this is the model of the growth of an actin filament. So we have the plus end on the right, minus end on the left. Notice on the minus end, there is also some additions of new components. No, but as the concentration of monomers drops, there is more removal of components in the minus end than adding. But the adding of components in the plus end continues to be greater than the removal of components. No. So how do we call this model of the growth of actin filaments?
The treadmilling model, sir.
All right. No, so based on look at this, no, the new components are the dark orange. So look at them. It seems like they are moving along, no, the bands of a treadmill towards the negative end. So treadmilling model. So just remember, no, the difference. It's possible to add a new component in the minus end, but a lot slower, no, in actins. Because actins are also polar, there are certain motor proteins, no, that are involved, and they are the myosins. So the most common of these are the myosins found in our muscles, the myosin 2, which can actually attach tail to tail, no, so that we have these myosins that are back to back, bipolar in nature. And if you're familiar now in your zoology class on muscle structure, we have here in the bottom the, what's tawag ani? Unit. Kanina ay
The sarcomere, sir.
sarcomere. Okay, so we have here the actins, no, kaning or yellow orange, and then in between are the type 2 myosins nga ga back to back ang arrangement. So you can see the heads projecting here at the each side, and in the middle you can see the stalks, no, that are attached together. So this is the most common type of myosin. Um, and there are also unconventional myosins. So here, myosin 1 is used to attach, no, the actins to the surface of this microvillus. And then here we have myosins 5 and 6 that have similar functions to kinesins and dyneins, no, respectively. So here we have the muscle cell. The muscle cell is actually a very long cell. It's also known as the muscle fiber. And inside the muscle fiber are smaller fibers still called myofibrils. And the striping pattern is because of the sarcomere, no? So ang sarcomere kaning isa ka ani sarcomere unit. So we have the myosin 2 in the middle and the actins on each end. And with the help of ATP and the formation of a cross bridge between the myosin 2 and the actin, your muscle sarcomere can actually contract. And if they contract all together, many of them, your muscle will also contract. Okay, so that's the mechanism. So lastly, no, before we end, can anyone explain how um, or what's happening in this illustration? We start here with a neuron um, transmitting a signal. The signal reaches the muscle cell. What happens next?
Um, Sir, once action potential reaches the muscle cells, sir, uh, the sarcoplasmic reticulum then releases calcium ions. Once the calcium ions are released, they attach to troponin, which are also unsay tawag ni? Attached to tropomyosin. And when they attach, the tropomyosin sort of reveals these myosin binding sites found in the actin filaments, sir. And when those are revealed, of course, the myosin forms a cross bridge into the actin filament, and that's where muscle contraction happens, sir.
All right, so that's correct now. So signal from the neuron will first pass through the transverse tubule first, T tubules, and will stimulate the release of calcium ions from the sarcoplasmic reticulum, which is not really different from the ER, but they specialize in the storage of calcium. And you mentioned the mechanism here, the calcium binding to the troponin, which is in turn attached to tropomyosin. And when that happens, the tropomyosin gets out of the way, revealing the myosin binding sites. And the myosin is able to make a cross bridge to the actin. And with the help of ATP, can move the actin so that the sarcomere will contract. No, and then you have muscle contraction. All right. So I will not anymore review on the cell movement. Now I think you can just review this for yourself. Gamay lang man eh. Instead, just announcements for the semifinal exam, which is on March 31. Chapter 7, 8, and 9 will be the coverage. It's multiple choice. Minimum 50 items. It could reach near 100 items, no, na multiple choice questions for the exam. So that's it for this video teleconference. No, um, do you have any classes after this or any activities?
None ra sir pero nagkan-kuan ka po. Activities.
Daganimon.
Yeah.
Daganimon. All right, don't worry, I will not extend. Um, again, no, kamo na lang bahala mag-review atong cell movement. No, actually, kaning topic dire, kaning cell movement is more on actin. No, so you can see here integrins and then actins that are helping in the sticking of the cell onto the surface and with the formation of the lamellipodium para makamove ang cell. So this is the mechanism. The actin filaments are sort of branching out, no, to form the lamellipodium, as you can see in this electron micrograph. And this one is just a colorized picture of the cell showing the locations of actins and myosins. And of course, here on the bottom right, you can see focal adhesions, okay, along the cell. Okay. And then here we can see the formation of neural plate and then the neural tube eventually helped by the microtubules, no? The microtubules are the blue ones that help the cell become columnar. And then the microfilaments or the actins are the one here in this edge that help the cell become wedge-shaped. So wedge-shaped na ang cell mo, ano yung muluko dayon, magluko na dayon curve nga makita nimo. And eventually the formation of a neural tube as we see here. And in prokaryotes, they don't have those actin, microtubule, or intermediate filaments. They have equivalents, no, that have similar functions, and these are their names. So, mao man to. So, are there any questions or concerns? Time check 9:03.
Dili ra need og exam permit sir.
No, you don't need an exam permit. Okay, napay other concerns, questions? Okay, so if there are none, and if you have a question later on, just message me in eLearn, okay, or through Messenger. So again, guys, thank you for joining in this video teleconference. May you have a productive day ahead, okay, and you are now all dismissed.
Thank you, sir.
Thank you
Thank
sir.
you, sir.